Welcome everyone. I'm Rain Brown. I'm Program Manager with the Western Region Homeland Security Advisory Council. Thank you all for being here today, and we hope that you find this session informative as you go about making your decision of whether or not to receive the COVID vaccine and to learn more about some of how it works. And before we get started, what we thought we would like to do is get a sense of where you are right now and you're leaning towards whether or not you're going to receive the vaccine if it's offered to you. So I'm going to just launch a poll, and if you could participate and let us know what your current feelings are about taking that vaccine if it's offered to you. And I'm going to just leave that open for a few minutes while I introduce our speaker today, Michele Keane-Moore, who is a senior lecturer at Western New England University. Michele was looking for ways to help out in the region regarding the COVID and anything about the vaccine. So we came into conversations and thought that it might be good to offer these informational workshops to first responders and those folks who are in the first phase of receiving the vaccine. And so I'm going to pull it. Let's end the poll, and then I'll share the results and kick this over to Michele. And Michele, you're muted, just so you know, and welcome you to this session for first responders.
Thank you, Rain, and thank everyone for being here this morning. I am going to share my screen, pull up my slides. Good morning, everyone. I am Michele Keane-Moore, and I am going to hopefully present some information to you that you will find useful in making that decision when you are offered the SARS-2-CoV vaccine, which hopefully we will all be given that opportunity within the next few weeks to months. So I put together a list of questions I thought would be helpful in trying to decide whether or not to receive vaccine. One is, why do we already have vaccines against COVID, given that we're only about a year into the pandemic and since the virus has been circulating? How do these vaccines work? How have these vaccines been tested? What is still unknown at this point, and how will we get this information going forward? And what additional safety measures are in place for people who are getting vaccinated? So to answer the first question, we already have vaccines available because of previous research that's been done, and also because of Operation Warp Speed. So we need to go back to understand how these vaccines have been developed, so what their evolution has been. And the first thing to know is that it has been only since January 11th of this year that the genetic sequence of the virus that's responsible for COVID was identified and published. That information allowed scientists to be able to get the information that they needed for what has been identified as a protective target, a neutralizing target for the SARS-2-CoV virus that you see depicted here on the right-hand side of my screen. And the important thing to see in this picture is the red knobby protuberances that are coming out from the surface of that viral cell. SARS-2-CoV is a beta coronavirus, and it is these the molecules that are protruding, which are the pre-fusion spike protein trimers, or as they're referred to as spike proteins, that allow the virus to attach to our human cells, and they attach to a surface molecule on our cells called angiotensin converting enzyme 2, or ACE2. And by attaching to those molecules, it allows the virus to get into our bodies. So it was information that was gathered based on 2 other very similar viruses, namely the Middle Eastern Respiratory Syndrome virus, or MERS virus, and also the original SARS-1-CoV virus that allowed scientists to identify and narrow down that spike protein as a potentially good protective target. And in fact, all of the vaccine candidates either contain spike protein or the genetic information for spike protein. It is thought to be the most important neutralizing target for the immune response. So the other part, in addition to having information on hand, Operation Warp Speed was announced in May 15 of this year. The current administration decided to put support behind programs so that we could accelerate control over the pandemic. And as part of that program, they wanted to particularly advance development, manufacturing, and distribution of vaccines to help accelerate control over the pandemic. They had strict criteria for selection for different vaccine candidates, namely the manufacturer of each candidate vaccine would have to already have in hand preclinical study information to show that their vaccine had the potential to be protective in human beings based on animal models. Their vaccine candidate would have to be able to be scaled up to produce 100 million doses by mid-2020. And they would also have to have the ability to enter a phase 3 pivotal trial, a trial in human beings designed to test the effectiveness of their vaccine candidate. They'd have to enter those trials between July and November of 2020, and that's just to start the trial. And they had to adhere to one of 4 platforms. They would either have to have a genetic vaccine platform, a non-replicating virus vector platform, a recombinant subunit adjuvanted protein platform, or an attenuated replicating live vector platform. And the platforms 3 and 4, all of the vaccines you've received in your life would fall under categories 3 and 4. Categories 1 and 2 are newer and more novel platforms. So I will describe those in a little bit more detail because they're also they represent the ones that are farthest along and the ones that in all likelihood we are being offered now and will be offered first. So on December 12th and 18th, emergency use authorization was given for the Pfizer and the Moderna vaccine candidates that are based on a genetic platform because the basis of their vaccine is a messenger RNA. And I'll explain how that works in just a moment. And the third most likely vaccine candidate that we may see within the next couple of months would be based on that second platform of the non-replicating viral vector platform and might either be produced by AstraZeneca. This is the AstraZeneca Oxford vaccine. And the basis for that is a chimpanzee adenoviral vector, or equally Johnson & Johnson's vaccine candidate is based on a uncommon human adenoviral strain 26. And and is a similar platform to the AstraZeneca platform. And right now it looks like they may have information to support emergency use authorization as early as late January or February, and which would mean that it might make its way into being offered to a larger part of the public as early as March. There are many other promising vaccine candidates, and before the pandemic ends, it may be that we will see many more of these come and be offered, but because of the fact that Pfizer and Moderna are already out there, and either the Johnson & Johnson or the AstraZeneca is likely to follow, I'm going to just focus on those today. So what are messenger RNA virus or messenger RNA vaccines and viral vector vaccines, and how do they work? Messenger RNA vaccines have been in development since the 1990s. Prototype vaccines have been studied in animal models since 2005. The first time an mRNA vaccine went into human beings was a safety trial, and it was a rabies vaccine manufactured by a company called CureVac, and it was given to 101 healthy human subjects over the years of 2013 to 2016. More recent advances within just the past 2 years have improved the stability and the delivery of messenger RNA vaccines, although there are no licensed mRNA vaccines to date. So in order to understand how a messenger RNA vaccine works, you have to know first how proteins are made within our cells. So on the right-hand part of the screen, you will see a representation of a human cell. And within that human cell, a compartment in the center of it that contains the genetic information for not just how this whole cell functions, but the genetic instructions for our entire body in the form of a DNA molecule. So our genes are made up of DNA, and DNA has the DNA within our cells has all of the genes that encode all of the instructions for our cells and for our bodies. But in order for that information to be put into a functional form, what has to happen is a process called transcription, by which the information in just one of those genes gets put into a new form of genetic material called messenger RNA. And that message then leaves this compartment called the cell nucleus and goes into the cell cytoplasm, where the cell can then undergo a process called translation to make proteins, and proteins are the functional form for everything that happens within the cell and within our bodies. And there are two important things to know about this. First of all, our cells needs change. It's all the time. It's a very dynamic situation. So the messenger RNA is a very fragile molecule, and they are constantly being made and being destroyed very quickly by the cell, so that the cell can make the proteins that it needs just at that point in time. And it's also the fragility of this molecule, the messenger RNA, that is why the vaccines that are made using it have to be kept at those ultra cold temperatures. So that it is a good thing that the message is actually fragile, because the other thing to know about it is, after you're given that vaccine, the messenger RNA after it enters the cell and makes the protein that it has the genetic instructions to make, is only there for a short period of time. This doesn't last in your cells for a long period of time, and it doesn't last in our bodies for a long period of time. And I can show you that in this figure, which is basically the messenger RNA that's being used for these vaccines encodes that spike protein, which you'll remember from the picture of the virus is that knobby part that helps it attach and get into our cells. So the genetic instructions for that spike protein are encoded in this messenger RNA. And in order to help stabilize that message and help it get into our cells, it's given a lipid coat, also called a lipid nanoparticle coat. And again, that lipid coat helps to protect this messenger RNA. So when it's given as an intramuscular injection, it can get to our body cells, and that lipid coat also helps it enter our body cells and once it's in our body cells in the cell cytoplasm, that messenger RNA then turns our cell into a little protein factory for that spike protein. And that spike protein then can be seen by our immune system. So typically what will happen is that spike protein will be taken up by the cells in our body that are responsible for initiating an immune response called antigen presenting cells. And it will put that spike protein in a way that will make it possible for our T cells and our B cells to be able to recognize and respond to the viral spike protein. And when our B cells see that spike protein, what they're able to do then is make those neutralizing antibodies so that if the virus comes into our body, those neutralizing antibodies stick onto the virus and keep it from infecting our cells. So this is the process by which the vaccine works to generate a protective immune response. If you were given a virus vector vaccine, it works on the same principle, except instead of having the messenger RNA encoded encased in a lipid coat, what happens is they take that same genetic material and they put it into a virus. In the case of AstraZeneca, their vaccine uses a chimpanzee adenovirus that is unable to cause human disease, but is able to attach and help that genetic material, the spike protein genetic material, get into our cells, again, to turn them into protein factories for the spike protein and generate that protective immune response so that if we encounter the virus, we already have a protective immune response on board, those neutralizing antibodies that attach to the virus and keep it from getting into our cells. So the vaccines that make or have spike protein, like putting up most wanted posters all over the body, train the body to be able to look out for, capture, and kill any invading SARS-CoV-2 virus. So that gives you sort of the platform in the background to understand how these work. Does anyone have any questions at this point about any of what I've currently described? Okay, and you can always also put a question in the chat for me as well, but I don't see anyone having questions at this point. So I will move on to the next part, which is how these vaccines, how have they been tested? So these vaccine candidates have actually already undergone extensive testing. Typically with vaccine clinical development, and certainly in the case of these of the Moderna and the Pfizer vaccines, and the other candidates that would, if they got to the point of being offered to us, would all undergo these stages of development. The first stage of vaccine clinical development would be a phase 1 safety trial. In a phase 1 safety trial, what happens is a small number of healthy volunteers, usually on the order of just tens of people, will be given a dose of the vaccine, and they will be monitored for any safety events that might result from having received that vaccine. In the second phase of study, what happens is a slightly larger number of, again, healthy volunteers are given different doses of that vaccine candidate. And in addition to looking at safety, they are also, in the case of these vaccines, blood samples were taken to look at the antibodies that were being generated to the different doses. In the third part of vaccine development, vaccine candidates are evaluated for their ability to be effective, not just in terms of the antibody levels generated, but their ability to protect against the actual in the case of these vaccine candidates, being able to protect people against coming down with symptomatic COVID symptoms and disease. So for the Pfizer and Moderna trials, a very large number of people, between 20 to 40,000 people, were given, and these were not healthy volunteers. These not just healthy volunteers. These represented people across a broad spectrum of health backgrounds, ages. They tried to enroll people who would be representative of people who would need and be receiving the vaccine. And so this large group of people were divided into two categories, and they didn't know which category they would be in. They would either receive vaccine or they would receive a placebo saline injection. And for these trials, because it wouldn't be ethical to give people virus, we don't have treatments, and typically that isn't, people are not typically challenged in vaccine trials with virus. What happened is they tried to recruit people from parts of the country where COVID transmission was high, and or they were in a high-risk profession, such as frontline healthcare workers, where in the natural course of their day, they would be at a higher risk for encountering virus. And then after they waited, after either being vaccinated or being given placebo and told to go about their ordinary lives, what happened is they counted the number of symptomatic COVID cases and then compared how many cases occurred in the placebo group versus how many cases appeared in the vaccine group. So when they were looking for evidence for the effectiveness of the vaccine, what they are doing is they're comparing, again, the number of cases for people given placebo versus people given the vaccine. So for both Pfizer and Moderna vaccines, they have undergone phase 1 safety trials, phase 2 dosing trials, and now they are part of the way into their phase 3 efficacy trials. But what happened is they had already designed the trial also to say that after a certain number of cases were seen, and that number was about 200, that they'd be able to do that comparison and see whether or not there was evidence for their vaccine working, and that would form the basis for being able to come into the Food and Drug Administration and ask for emergency use authorization, where then that vaccine candidate could be offered for a broader use. And that phase 3 efficacy trial will continue through the next 2 years and will form the basis so that Pfizer and Moderna can come in and ask the FDA for full licensure. So what is an emergency use authorization? It's not a fully licensed approved product. It's at an earlier stage, as I just showed you. However, there is information available based on those phase 1, 2, and preclinical trials as well, and those first 2 months of the phase 3 trial, to say that there is a reasonable safety profile with both of those vaccines. And they have also both demonstrated efficacy based on risk reduction in vaccinated subjects. So of those 200 cases, most of those cases were in the people given the placebo and not in the vaccine group. And when you do the calculation, turns out that that's about a 95% risk reduction, which is very high. The FDA had actually set a much lower bar for that. So overall, there is a favorable risk-benefit profile given the pandemic circumstances. If we weren't in a pandemic, they would not be looking to grant emergency use authorization. But because being in a pandemic increases people's risk, they do take that into account in deciding to grant an emergency use authorization. However, those phase 3 trials for licensure will continue through the next 2 years and will hopefully generate information to support a fully licensed product. So what is known at this point based on the information in both the Moderna and the Pfizer trials? When they did that calculation for efficacy, again, they found about a 95% efficacy against symptomatic COVID for at least those first 2 months of the trial. They found in people who were vaccinated, in the blood samples that they have taken from these people, that neutralizing antibodies that are equivalent or higher than people who have recovered from natural COVID infection, also known as convalescent human sera. So the level of neutralizing antibodies is very encouraging. There's also preliminary evidence that Moderna's vaccine is protective against development of severe COVID. The reason why they were able to do that analysis is, again, by comparing the placebo group against the vaccinated people group. They found that there were 30 cases of severe COVID in the placebo group and no cases within the vaccinated people group. What is currently unknown, and is important to know if you have received or do receive one of these vaccines, is that we don't know yet if by receiving the vaccine, if you will be protected against asymptomatic COVID infection. If you were to have an asymptomatic infection, you might potentially be infectious to others. So if you receive one of these vaccines, you will be told that you will need to continue to mask and socially distance and protect the other people in your life, just in case, again, you're able to be asymptomatically infected. They are very actively looking at that question, and we should know more information soon as to whether or not people can be asymptomatically infected who are vaccinated. We don't also currently know how long this protection will last, but that's also true for people who have recovered from natural COVID infection. It's just, it is one of those things that is also actively being studied. In the information that both Moderna and Pfizer came in with to ask for their emergency use authorization, most of the cases that they saw occurred in Caucasian people, even though there are a lot of there is a lot of diversity. There's still the majority of people participating in the trials were Caucasian. Most of the cases occurred in those Caucasian groups. So at this point, we don't have enough information to really know what the efficacy will be like for non-Caucasian people. However, that being said, there isn't any reason to think it would be very different. And in looking at the number of cases versus that have occurred in placebo versus vaccinated people, it seems like if they look at the different groups, that the numbers are being consistent with the larger group. But again, this is an area that is actively being looked at and will be followed going forward as they collect more information and cases in their phase 3 trials. And at least for the Pfizer, again, just because they didn't have that many severe cases in either the placebo or the vaccine group, the evidence for protection against severe disease, the data are still limited, and all of these things are actively being tracked and studied going forward. FDA has an additional rationale for being able to grant an emergency use authorization after 2 months of collecting of data, and that is historically uncommon but clinically significant adverse events plausibly linked to vaccines generally have an onset within 6 weeks following vaccination. And so they now have 8 weeks and more going forward. So they feel like common and uncommon severe adverse events have been actively looked at and monitored for, and it is still possible, because when you go from vaccinating tens of thousands of people to millions of people, rare events can come up. But I will describe the safety net that has been put into place to look for any type of rare event that could come up. Does anyone have any questions at this point about anything that's been presented?
I do.
Okay.
So my name's Jen. I've been a paramedic for, 25 years, but I also worked for Pfizer in phase 1 clinical trials. So, and we used to do phase 0 clinical trials on animals prior to phase 1. And I know for these trials, they didn't do any animal trials. They went straight to phase 1. What's interesting is in phase 3, for the 20, to 30,000 that you have listed, the clinical trials I could find were 44,000 people that they tested on, but only 36,000 finished the 2-month trial. So that's 20% didn't finish. And I don't think that's out there, very high in the public, that 1 in 5 people didn't even finish this vaccine trial.
It may also be because, don't forget, that 2-month interval, they had additional criteria, which was that people had to be a certain length of time after getting their second dose. And so they were enrolling and vaccinating people all the way along there. And so I think because the attrition from the trial, the FDA Vaccine Advisory Committee, they had the numbers in terms of the number of people who started the trial and who are still enrolled in the trial, and they haven't decreased 20%, but the analysis probably was not done because not everybody had enough length of time by the time that they had accumulated that number of cases in the trial to do their analysis, so that there are more people. They just simply weren't part of that analysis because they didn't have the same length of time in yet. Does that make sense?
No.
Okay.
No, I guess not. I mean, because when we did the clinical trials at Pfizer, and we would sign someone up, and we counted them at the end of the day to
Yes.
say, you know, this is how many people we had in the trial at the beginning, and this is how many people we ended the
right,
know.
but they're not, my point is they're not done with the trial yet, so they're still enrolled. And I can actually get you those numbers.
Okay.
Because they were part of the public information that was given to the advisory committee so that everybody could see it. They've really been trying to be very transparent in this process.
Yeah.
And
go ahead.
I can very much get you those numbers, but it hasn't been a full 20%, and even in each individual category, they've actually had a very good retention of people within the trial.
Oh, okay. Because it just seemed like the side effect blowout was a lot higher than is being reported. Okay. All right. Thank you.
Great.
That was my question.
Excellent. Thank you. Would anyone else like to ask a question at this point?
Michelle, there's a question in the chat there. I'm not sure if you saw it or if you were able to answer it. And the question was, does this mean the vaccines will work on any variant of the COVID virus?
Oh, that's a great question. And it is another open question at this point. So there isn't just one variant that's circulating out there. The virus has been changing because it's what viruses do, and particularly the SARS-CoV is an RNA virus, and they tend to collect more changes than others when they make a lot of themselves. So there isn't just one variant. I know there is the variants that have come into the news most recently, and particularly the one that has a deletion mutation, so that they think that that is in the spike protein, and they think that it may be more transmissible among people. They are currently actively looking to see whether or not the neutralizing antibodies that are made will neutralize that both in vitro, just looking basically at experiments with cells in culture, but also within animal models as well. So we don't fully know the answer to that, but I think that they are feeling confident at this point that the vaccine will still cover those variant strains. Once a very large number of people are vaccinated, there'll be a lot of pressure on the virus. There'll be selection pressure to select viruses that escape that, and we'll probably have to deal with that at that point. But at least at this point, it is likely that the vaccines will be able to be protective. And I have not seen anything that would suggest otherwise at this point. And I know that most of the fight that Pfizer, Moderna, and AstraZeneca, as well as other manufacturers, are actively looking to see for their particular vaccine candidate how it will respond with different variant strains. All right, so, and there will be opportunity for questions at the end too. So, and this gets to maybe something that Jen was touching on too. How may you feel after receiving an mRNA vaccine? Based again on the information that they've collected from the trials and also what people are finding now that they're receiving them in the larger public. For most people receiving it, your arm will hurt. 2/3 of you will feel tired after receiving it. Half of you may have a headache. 1/3 of you may have muscle aches and chills. 1/4 of you may have joint aches. 1/6 of you may run a fever. And the important thing to know is the likelihood of feeling this way will be higher after your second injection rather than the first. And the younger you are, the more likely you are to experience these. And the reason for both the fact that it's more likely after the second injection, and why if you are younger, you're more likely to experience these is these are all things that happen when your immune system is responding to something. And so although you may feel bad, these are all actually signs that your body is doing the right thing and making a good and vigorous immune response against the vaccine. FDA will continue to collect information through the phase 3 trials and through the emergency use authorization period. Additionally, there is safety monitoring that will occur for everyone who is receiving vaccine. The first way that this will occur is a collaboration that takes place between the Center for Disease Control and the Food and Drug Administration. It's a program that's been in place for years called the Vaccine Adverse Event Reporting System. However, with the rollout of Pfizer and Moderna, they've added on to the reporting system a text-based monitoring program. So if you receive the vaccine, you will be asked to participate in this program. And if you want to, what happens is that they will send you a text every day and ask you how you're feeling and collect any type of symptoms you may be having after receiving the vaccine. That will go into a national database that will be available to the public, and it's designed to detect those rare events that might signal that there is a safety issue with the vaccine. There is also a vaccine safety data link program. If you are one of 20 million people who participate in an integrated healthcare system, such as Kaiser Permanente, they will also be looking on a weekly basis, so nearly in real time, to see if anyone receiving a vaccine is experiencing any kind of a safety issue. There is also a clinical immunization safety assessment with some participating medical centers, including our Boston Medical Center, who are collaborating with the Center for Disease Control to both answer questions that US healthcare providers may have about whether or not their patients should receive vaccine, but we'll also be conducting independent clinical research as to the safety and effectiveness of these vaccines. And lastly, there will be special group monitoring through the Vaccine Adverse Event Reporting System for special groups such as pregnant women and long-term care facility residents. So as an example of how safety monitoring is in place and working, the United Kingdom started actually vaccinating people with Pfizer's vaccine about a week before we did. Within that first day, 2 healthcare workers were reported with severe allergic reactions after receiving their first injection. And the question was asked, are these related events? And as a precaution, the United Kingdom recommended that people with a history of severe allergic reactions, anyone who carries an EpiPen, to not receive the vaccine, and also to receive, for people receiving it, to only receive it in a facility with resuscitation equipment. On the first day that the US rolled out Pfizer's vaccine, 2 healthcare workers in Alaska were identified with severe allergic reactions, and one of them did not have a known history of allergy. There has been 1 report of an allergic reaction to Moderna in a person with a history of shellfish allergy and who carries an EpiPen. Currently, the recommendation is from FDA and through the CDC is to that vaccines should be done where an EpiPen is available and anaphylaxis can be managed. So it's not, if you have, unless you have a known allergy to one of the components of the vaccine, people just with a history of allergies are not being excluded from receiving the vaccine at this point. However, anyone with a history of allergy is also encouraged to, instead of staying for 15 minutes after receiving their vaccine, to stay for at least 30 minutes in the facility where any type of allergic response could be managed. So these additional safety monitoring programs will provide important real-time information to vaccinated people and allow them to stay informed. There are also circulating misinformation and myths out there on the internet, and I thought I would just address several of these that I've seen. The first was that mRNA vaccines use aborted fetal tissue for their production. And the fact is, messenger RNA vaccines don't use any cells for their production. It's a completely cell-free, animal-free synthetic process. The second is mRNA vaccines can cause your DNA to become mutated. And the fact is, if you remember from my slide describing how proteins are synthesized, messenger RNA is an expression product of your genes. It's made differently than DNA, and it can't combine with DNA. Also, any messenger RNA that enters from a vaccine would only go into your cell cytoplasm, not into the nucleus, so it's not even near your DNA. And then the third thing that I've seen is mRNA vaccines have more side effects than influenza vaccine. And in fact, these mRNA vaccines do seem to produce a greater degree of vaccine immune reactions, such as the redness at where you receive it, pain at that injection site, muscle aches and fever, especially after the second dose or if you're younger. And so if you are a team leader, you may not want all of your team to be vaccinated on the same days in case people do have a day after or so when they're vaccinated if they don't feel very well. So all of the decisions we make are always looking at the risks and weighing them against the potential benefits. So I don't have to tell any of you on the front lines how unpredictable and potentially harmful coming down with COVID is. The virus itself has been shown to have short-term and potentially long-term negative impacts on our lungs, heart, kidneys, and brain. It may be that certain vaccines might be best suited for particular individuals. In a perfect world, we might be able to look at all of the vaccine candidates and decide which one would work best for us. But in all likelihood, that's not going to be the way that these vaccines are offered to us. However, Vaccination, I feel, is our best defense against this virus and may help us turn this pandemic around. But obviously, the decision is up to each of you as to whether or not to receive it. I've included a table that gets into a lot of detail comparing what's known about the Moderna versus the Pfizer versus the AstraZeneca vaccine candidates. And I offer that for your reference. And I would be happy to take any questions you have.
Michele, there is one question in the chat that came up and saying, I'm assuming after being vaccinated, you won't test positive on a PCR COVID test.
Right, so that is true. You shouldn't test positive on a PCR test because the messenger RNA would not be in your body that long. And when they do a PCR test, they are looking probably in, I think that they look for a different part of the virus rather than the spike protein gene. So there shouldn't be any of a cross reaction with that. If they looked by antibodies, you might come up positive on an antibody test, but only if they were looking specifically for spike protein antibodies. I know that within the context of the two trials, one of the ways that they are looking to determine whether somebody is positive for if they became infected with COVID during the trial was they would look at antibodies against a different part of the virus. And they were also able to use they were definitely using the same tests that are out there to look to see whether anyone had virus by PCR. So I think the answer to the first part is no. And if you looked by serology, if it was a test that didn't include spike protein, you shouldn't come up positive on that either.
There's another question in the chat regarding the antibody test, and after receiving the vaccine, can one do an antibody test, and how long after getting the vaccine can an antibody test be done?
So you'd want to know whether or not you had made a good response to the vaccine. I would guess that the way that the information was presented to the FDA's advisory committee was that it showed that protection starts to come on board after your first vaccination, but it doesn't fully come on board until at least about a week to 10 days after your second vaccination. And that's when your antibody levels start to really get high. So I think that they feel that people are partially protected after the first vaccine, but definitely within about a week of your second vaccine, you should be fully protected. And as such, you should be able to see your antibody levels on a test. We don't know yet how long the antibody levels stay up following vaccination, both in people who have recovered from natural COVID infection, as well as people who participated in the earlier parts of the vaccine trials with these two vaccine candidates, found that the antibody levels over time do come down, which is natural and normal. However, they also found that people were able to generate what are called memory cells. And those memory cells would be that long-lasting type of protective immunity that help you respond if, for instance, you didn't encounter the virus until a year or longer after you got vaccinated. So generation of memory cells is very important, and there is preliminary evidence that those cells are generated to the vaccines.
Thanks. We got a couple more questions that have come in. One is, now that there are 2 manufacturer vaccines are available, would you be able to choose which one, and is one better than the other at this time?
I don't think most people will be offered a choice. I think because of the way that people are coming up within priority categories, and we have at this point more people who need or who would like to take the vaccine than we have vaccine. My guess is, for someone who feels like they would like to receive a vaccine, if you get the opportunity to take it. And in terms of, what was the second, I'm sorry, the second part of the question?
Is one of them better than the other?
Fine. They are both really efficacious. When the data were presented to the FDA's Vaccine Advisory Committee, there was not a clear-cut difference in terms of the levels of antibodies being generated and the protective nature of those antibodies. I would say with Moderna, again, because of the fact that they had severe cases of COVID in their placebo group, they were able to address that question, and they didn't have that in their vaccinated people group. They were able to at least offer preliminary evidence that they protect not just against symptomatic, but against severe disease, and that's encouraging. I didn't see anything that would make me think that Pfizer would be different than that. I think that it's a very good question, and we will probably know more going forward about if there are differences and if one is really better than the other. They are different from each other. They are a little bit different in how they present how they take the genetic material for the spike protein and give it to people. So they're not identical vaccines. And they may in fact have differences that we'll see going forward. I think it's just simply too soon right now to know if one offers a clear-cut benefit over the others, if one is better.
And another question in the chat is, because reactions to vaccines appear to include some of the same symptoms as the COVID virus, should we keep anyone with those symptoms following administration of the vaccine out of work until those symptoms are resolved? I know that
that question has definitely come up, especially within hospital systems. And I think that, and I will have to, I know that hospitals have put into place policies to address that particular question. And I don't remember exactly how they, for some reason, I'm thinking that it was a certain length of time after receiving your vaccine that you're more likely to have those symptoms. And I think because of the fact that they do overlap, that hospitals did put into place policies that would say that if you are experiencing those symptoms, that you should not report to work for a certain period of time. But I will have to look that up and get back to you in terms of what this specific recommendation was. But you're absolutely right, they do overlap.
Next question is, do you see a concern with the temperature issues with the Pfizer vaccine? The sub-zero temps are difficult to maintain in transit versus Moderna's vaccine, which does not require the same temperature control. Couldn't the instability in temperature cause a problem with the effectiveness of the vaccine?
Yes, it certainly could. I know that from everything that I have seen and read, though, that they have temperature monitors put into place. They have things so that they can detect if the temperatures go out of specification. And so in all likelihood, what that might result in is more of their vaccine going to waste or not being used if it falls out of specification, rather than receiving a vaccine that they were not able to detect when out of specification. It is an issue. It's been identified as an issue in terms of the fact that it has to be kept in that temperature range, but they also put into place, because of that, extensive monitoring to make sure that that people don't receive a vaccine that has gone out of specification. They also, in terms of where they are distributing vaccine, have tried to limit the Pfizer distribution to only to facilities that can keep that have the ability to keep it that cold. And I think they're directing Moderna to facilities that would have a greater issue with that.
And then a question is, do we have any indication of how long a period of time the vaccine offers one protection from COVID-19?
We don't know that yet. And again, that will be one of the things that they will be monitoring very closely going forward, both within the context of those phase 3 trials, and also just generally speaking, anyone who receives vaccine, particularly if you choose to participate in any of the monitoring, the Be Safe, or if you're part of a larger healthcare system that is doing that monitoring, they will definitely be looking at that question very closely.
Thank you. So that's all the questions that are in the chat. I'm going to go ahead and put forward another poll. If this is one regarding, do you feel that the information from this session has helped you decide about taking the vaccine? We'll let people go ahead and fill that out. If you do have a question, again, feel free to unmute yourself and ask the question or put it into the chat.
And people are also free. My contact information is on the first slide. And you are very welcome to send me any questions that you can have, through my Gmail email account, and I will be very happy to answer them and help you get the information you need.
Thank you for that. So most of you either strongly agree or agree that this session gave you the information you needed to help decide. And then I'm just going to re-ask the same question which we asked the first time, and where you're at in your willingness to get a vaccine. And we're just using this information to help us assess these sessions and where folks are at. And like I said at the beginning of the session, we will be sharing the slides out with everyone who attended today. So you'll be receiving these. And again, that will have Michelle's email there if you didn't get that at the beginning of this session. So you can go ahead and contact her with any other further questions that you might have, or questions that might rise up after you've had some time to think about this. So feel free to contact Michelle and or myself with questions, and I'll make sure that to get them to Michelle. We do have one more session of this coming up on Wednesday at 2 o'clock. I'll send the information of that out when I send the slides out. So if you have colleagues or friends who might be interested and benefit from this session, they can go ahead and register for that for Wednesday. And then this has been recorded, this session, and I'll be working on getting that up on the Homeland Security Council's website. So the results from the final poll are there. and there's pretty much where we were at the beginning with people's sense of whether or not you're going to be taking the vaccine or not. So, Michele, any final words?
No, I think that everyone should think about this and make the right decision for him or herself. And but overall, I think that vaccination is going to be one of those tools by which we can actually change the course of the pandemic.
Thank you, everyone. And please share information about this next coming session with those who you think might need it. Thank you.